we're ready for a little bit on anatomy, right? And again, not the focus, but we're going to point out the anatomy as it ties, because it's going to tie into the physiology that we're going to discuss. And so if we look up here, right, we've got, this is called a lung cast, this guy right here. That is a cast of lungs. So literally what they do is they take, a dead person or a dead animal, and they fill the lungs with a material, a liquid that hardens. And then what they do is they digest, like enzymatically remove, or using acids, remove the organic material, and you're left with this, usually some kind of acrylic, sometimes it's latex, but you're left with this cast of the inside of the lungs in this case. And so you can see the trachea here, right? And then you've got the main branches off that trachea, right? Those would be the primary bronchi. And we start counting out here. We've got secondary bronchi, and so on and so forth, right? And what you can see is that the branching of these bronchi is extensive, like way more extensive than you may have ever realized or learned. In fact, to get all the way out to the ends, and frankly, the alveoli are not shown here. The casting material is usually too viscous to get all the way out to the ends, right? This would be the very end of that respiratory tree, right? So in other words, this model, right? So this right here, I'm circling, would be sort of extend from right here, right? Here it is. Draw some alveoli, right? Because it just wouldn't show up in this cast. It's just too small. Okay? So massive branching to lead to, the purpose being to lead to 300 million individual alveoli. The number's not important. The magnitude is, right? The reason we need 300 million of them is to generate a surface area that is far bigger than if this were just a bag, in the shape of a lung, right? It would have a surface area 1/1000th of what it actually has. Okay? So, but through all this branching, we get this enormous number of individual alveoli, each of which is like a little bubble down here, right? This sort of epithelial sac, if you will, that increases the surface area. So it's kind of like, the lungs are kind of like a sponge with really small bubbles in it, right, to increase its surface area. And again, go back to Fick, it's enhancing diffusion, right? Now, everything you see, so these, what am I getting at here with this? Conducting zones versus the respiratory zones. And I'll lay this out in what's called the Weibull model in the next figure, right? The next slide, I believe. But I can point to it here as well, because everything you see in this cast is conducting zone, right? Because those are simply the pipes, the branching and branching and branching of the pipes to get you to the respiratory zone, which is what we have over here, right? The respiratory zone is where we're actually having gas transfer, gas exchange across that thin squamous respiratory membrane, right? The conducting zones are simply conducting, carrying the air to and from, right? Now, I'm going to jump ahead here for a second. We are tidal ventilators, right? Well, what does that mean? Well, I'm going to draw a really simplistic lung. I'm going to draw it over here on the white where we can see it a little bit better. If I draw a really simplistic lung, it's going to look like this, right? And we are moving air into and out of that bag, if you will, during ventilation, right? But the fact that we're moving it in and out of this blind-ended, dead-end bag, well, 300 million bags, right? These little individual alveoli, describes us as a tidal ventilator, right? Think of the tides. They go in, they go out, they go in, they go out, right? That's what the air is doing. It's moving in and out of this sort of blind-ended bag. There are other organisms that have a respiratory system that looks more like this. where you'd go in on one side here and you'd come out the other side here, right? Those are unit, right? This is one tube just sort of like folded back on itself. Those are unidirectional ventilators, right? So bidirectional ventilation versus unidirectional ventilation. Unidirectional ventilation, you're moving in one way and out a different way, right? It's all moving in one direction. Here you're moving back and forth, right? So you're moving in two directions, if you will, right, along the same pathway. And so there are limitations to tidal ventilation in that we do not fully collapse our lungs when we exhale. So when you take a breath inward, you do not bring fresh air all the way down into this bag because the bag already contains air, right? And so there's a mixing effect that goes on in tidal ventilators that does not go on in unidirectional ventilators. Birds have a lung that's much like this with the single one direction path. Fish, or maybe an easy example, right? You're used to seeing a fish sort of opening and closing its mouth, right? And what they're doing is they're pulling water, right? The respiratory medium for a fish. They're pulling water into their mouths, and then in between that sort of gulping of the mouth, they're sort of squirting that water across their gills and then out the gill slits along the sides of their head, right? And so it's moving in one direction, right? In their mouth and then out the gill slit across the respiratory membrane in their gills. It's a completely different pattern of the respiratory apparatus in those organisms. Okay, we'll come back to these limitations because we're going to talk about the gases, the gas pressures in the alveoli here, right? Because they're going to differ between here and the air that we breathe in that's out here. Okay. Now, to go back for 1 second, is convective airflow versus diffusion, right? Because if you think about it, we haven't talked about ventilation yet, but we will, and we kind of have the idea that we're moving air into, and then back out, but into these blind-ended spaces, and you think, okay, you're going to just pull air all the way down into the alveoli. No, you don't, because what's going to happen is the total volume, cross-sectional area of the alveoli is enormous. And so what happens is air moves really rapidly through the small cross-sectional area, tracheae and primary and secondary bronchi, but then it gets slower and slower and slower because it's moving into more and more of these, branches that are increasing the total cross-sectional area of the pipes. And so, air moves really slowly. Well, then you're moving, remember, these don't collapse when you breathe out. So you're moving air in here, and it's sort of like, well, do they just expand more? Well, what really happens is you pull air through convective, i.e. pressure-driven movement, right? Pressure gradient-driven movement, down to about here. And then the gases have to basically diffuse back and forth, right? And they do so very quickly because it's in air, and this is about a millimeter between the alveolar ducts and these the alveoli in a particular cluster here called the alveolar sac. And so that diffusion of oxygen into the alveoli or carbon dioxide out actually occurs quite quickly, right? So it's a short distance, and diffusion in air is much faster than diffusion in water because of the viscosity of the solution, right? Air has very low viscosity compared to water. Okay, one of the interesting little bits that kind of pointed researchers to this fact was smokers, and if you experimentally ventilate a lung with particulate matter, once the particulate matter gets big enough, it's large enough. Remember, molecular weight's in there. It won't diffuse, right? And so if you get to like you're breathing, like smokers breathe particulate matter. Well, some of that particulate matter is so large it literally won't diffuse. So if you look at a smoker's lung, there's not particulate matter down in the alveoli. Guess what? It's all piled up here in the smallest bronchioles. And I could go backwards as well. It's just easier to draw it right here, right? These little red dots I'm drawing is the particulate matter. It stops moving because that's where the convective airflow stopped. That was the first line of evidence that said, aha, look, molecules that won't diffuse don't make it down into the alveolar sacs where the gas exchange occurs. And whereas oxygen and carbon dioxide, small little molecules readily diffuse this across this distance, but the big ones pile up here, right? So if you look at a smoker's lung, the histology of it, you'll find all of this junk sort of piled up in the smallest bronchioles, but not much of it down here in the alveolar sacs. Okay, next. The Weibull model. All right, in the Weibull model is basically like the explanation of that last slide, right? It's got, here's our sorry. Here's our trachea, right? And it branches and branches and branches, right, in that conducting zone, right? The cast picture I had, right? All the way out to about the 16th division of branching. No gas exchange, right? It is all about carrying. Note the surface area. It's very cross-sectional surface area. It's really small, right? Okay, way out here at the 16th division, it starts to go up a little bit, right? Then you move into the respiratory zone. Now we're down in that picture on the right, right? Over this picture over here, right? These last series of branches down to the alveoli themselves. And look what happens to the cross-sectional area, right? Takes off, becomes enormous, okay? And that, again, is because of all these little bubbles that we've got here, the alveoli. And again, they actually butt off the sides of the smallest bronchioles as well. So we start to get gas exchange in this area, which is why it's called the respiratory zone. Sorry. Keep forgetting to grab my pen each time. Every time I switch slides, I got to reactivate the pen, right? But the respiratory zone, right, is where our gas exchange occurs because we have that thin-walled membranes of the alveoli that minimize the diffusional distance such that gas exchange will actually occur. Okay. Now, if you look at some of the characteristics of these two different zones, right, we notice the epithelium in the conducting zone is much thicker, much taller. These are columnar cells. Whereas down here in the respiratory zone, they become squamous, right? Squamous means flat. Flat because we want our diffusional distance from A to B, right, across that membrane to be really short, right? Delta X. Delta X over here is much higher, right? Diffusion would be lousy. Okay? Now, in addition, we've got some structures in this columnar epithelium that are protective, right? We have cilia, right? And we have mucus secretion. The cilia and the mucus secretion work together, right? The mucus being produced by these glands that we've got shown here, right? These are mucus secreting glands. They're actually mucus secreting cells as well. They're called goblet cells because of their shape. And that mucus produces a wet lining that traps airborne particulate matter, right? It could just be dust, like, dust and dander, like pollen and things like that. Even skin cells, like a lot of dust is actually human skin cells. It's kind of gross. But it's not necessarily like pathogenic, right? But this could include things like bacterial spores and fungal spores, which can be pathogenic. They could, right? They could set up camp and start to multiply and grow, and you don't want that, right? And so what happens is the mucus traps this particulate matter, and then the cilia moves the mucus containing the particulate matter, and it sweeps it toward, right? And so within these conducting zone bronchioles, the cilia sweep this way, right? The cilia sweep toward the upper parts of the trachea. Well, guess what's at the upper part of your trachea? Your pharynx, which is your throat. And so what happens is you sweep this mucus upward and then you swallow it, right? And it takes it and it puts it into the GI tract where it is readily digested by enzymes and acids in the stomach, so it destroys it, right? So it's a lot safer to have that particulate matter end up in the GI tract than to have it accumulate in the respiratory tract. One sort of interesting note is that nicotine paralyzes these cilia short term, but again, if you smoke a lot continuously, throughout the day, they stay paralyzed, which means the mucus isn't moving, which means you're much more susceptible to a mucus buildup. You get that smoker's cough. And B, that particulate matter gets to sort of hang out a lot longer. And so respiratory infections can actually go up. Okay. The last component down here, well, two of them actually. Cartilage in the larger pipes helps hold them open. Just when we're changing thoracic pressures due to breathing, we don't want those pipes collapsing. And then when we get to the smooth muscle, right, the smooth muscle is actually helping us, and it's really important sort of in this range right here, right, because that's what's going to allow us to sort of shunt air to and from different regions, small regions of the lungs, right, sort of maximizing the distribution of air to the distribution of blood in the pulmonary capillary system. Okay. And that smooth muscle was seen. We're going to lose our pen again. I won't go back, but it's seen in the right figure previously, right, where the small bronchioles have significant smooth muscle that allow that constriction or dilation of the small bronchioles. If you have asthma, you're sort of well aware of that smooth muscle, whether you knew it or not. But if this smooth muscle abnormally constricts, as it does in asthmatics, you get an increase in resistance to airflow. It becomes very difficult to ventilate the lungs. Okay, last question over here is, what is the anatomical dead space? Well, when we get to spirometry and we start talking about measuring lung volumes, this will come back. But I want to point out, anatomical dead space is the volume of the conducting zone, right? And it's called anatomical dead space because it's not functioning in gas exchange. Okay, it is, you ready for this? It is about 150 milliliters in this conducting zone, right? The total lung, like the respiratory zone has, 3,000 mils, 3,000 plus mils of volume, right? So the conducting zone, despite it looking very extensive, right? It is very extensive because of all this branching, really has a small volume. And that's good, right? Because you got to think of this as like a snorkel you're breathing through. It's not really doing you any good except for getting the air from point A to point B. You really want the large volume down here where the gas exchanges can occur, right? And that's absolutely true. Okay. Let's talk a little bit about anatomy of the alveoli themselves, and we're going to talk about a couple different cell types found down here. Right? And we've got our type 1 cell first. These are the cells, these are the squamous cells. They look like fried eggs, right, with the yolk part being the nucleus sort of bulging because the cell has so little cytoplasm. These are the cells that form the walls of the alveolar sac, of the alveolar wall, I should say. They are part of the diffusive barrier, right? We'll blow them up over here on the right in a second. We'll talk about that. Type 2 cells do not form part of the wall. Rather, they're sort of interspersed, but they're not really you're not really diffusing across them because they're big fat cells. These cells are producing something called surfactant, right? And surfactant is all about reducing the surface tension of the water that lines these alveoli, right? There's a very thin, that's not what I wanted to do. There's a very thin layer of water, I'm going to put it in blue here, that sits on the inside of the alveolar wall, right? This blue line is a thin layer of water sitting here, right? And in an upcoming slide, we're going to talk about the surface tension of this water. So keep this line in mind here, right? So the surfactant is breaking up, reducing really the surface tension of this of the water molecules in this lining of each alveolus. And that's important because it helps sort of minimize the collapse pressure of those smallest spaces in the lungs. Okay. Another thing I want to point out is the capillaries that are associated. I can circle one here, right? So this is supposed to be a capillary right here, right? This is another one over here. And I note that they're wrong, in this picture, because the reason I point that out is because without, you can have all the surface area in the world in the alveolus. If you don't have matching or nearly matching surface area of the pulmonary capillaries, it doesn't do any good, right? It has to be surface area of what's called the respiratory membrane. The respiratory membrane being this right here, the shared barrier between air and blood, right? This is actually the wall of the capillary on the right here, and then the wall of the alveolus on the left here, sort of sandwiched together. So the respiratory membrane forms the diffusional barrier, and its thickness would be what we call delta X in the Fick equation, right? And I've got, it's called different things depending on how people write Fick. Sometimes they use T for thickness. I think I might have originally done that and changed it in my newer versions of the chapter. Sometimes they use D for distance. I've started to use X fairly consistently. Okay, but if you look at this image down here, if you look at these, it almost looks like the alveoli, right? The alveoli have been removed. This is a vascular cast, right? And so that is the blood supply to a single alveolus, what I circled there in blue, right? Notice it's called sheet flow for a reason, right? The blood flow through these pulmonary capillaries, because it's almost like it's not running through individual little pipes, right? The pipes have almost kind of fused in many ways. There's these little gaps, little holes here where there are spaces between the tubes, but they form such a dense network that it's called sheet flow, the blood flow through those pulmonary capillaries. And again, because we're maximizing the exposure of the blood to the alveolar wall, because that's our effective surface area, right? It's not just the alveolar wall. It's got to be matched by surface area of the pulmonary capillaries. Okay. You'll note we've got a couple other things here. We've got these pores. Well, we've got to connect this alveolus and all the others to the ducts that feed air in and out of it, right? Again, largely through diffusion. We've got this cell called an alveolar macrophage here, right? So it's a third type of cell, or maybe a fourth type of cell. I forget how many I'm labeled at this point. Anyway, this alveolar macrophage is a white blood cell, right? This is a white blood cell type that sort of hangs out in lung tissue. It moves around through amoeboid movement, and it gobbles up the particulate matter that escapes that ciliary mucus defense system. It's called mucociliary defense, right? The system we were talked about with the cilia and the mucus. You don't want mucus down here, right? Mucus would increase your diffusional distance. Okay, we have this very thin layer of water, right? Water's more viscous than air. It slows down diffusion. You want really thin layer of water. You certainly don't want mucus and quadruple or increase this thickness by 10-fold or 20-fold or whatever it might be. It would dramatically harm the rate of diffusion. So we don't want mucus, which means we don't need cilia. And so that all ended, if you look at the Weibull model again, that all ended before you got to this respiratory gas exchange area. Right? Now, that doesn't mean that some particulate matter might escape that system, and so that's why we have the macrophages down here, helping you fight infections at the terminal lung. Okay? Last thing I'll point out on this one, I have this picture down here, right, showing what alveoli really look like. Everybody draws them as spheres, and they really aren't, ever. Because they're so flimsy walled, they sort of butt up. And this picture kind of helps. You kind of see that, it's kind of like flat walled on each side, right? Kind of hard to pick out, right? This one here, flat wall, flat wall, flat wall, right? So they're these flat-walled polyhedrons, largely, because they have flat sides where they butt up against each other, and they look like soccer balls, really, right? Where they've got these flat panels. Right? And that actually becomes important in an upcoming slide where we talk about the collapsing or potential collapsing of alveoli. Okay, I think we are good on this slide. Okay, let's get back to this idea of surface tension. And again, we're talking about anatomical things because we're talking about those surfactant cells, the type 2 alveolar cells, epithelial cells, the production of surfactant, and how it relates to surface tension, right? And now notice I say this potential bronchiolar collapse. I'm not going to really talk about alveolar collapse. I'll come back to that in a moment, right? We just said a moment ago, our type 2 cells are producing this surfactant. Well, why do we need it? Well, if you look over here on the right, this is a droplet of water, right? Kind of like I'm having this picture down here. Whoop, I forgot to grab that pen again. I'll get better at that. Okay, so here we've got a droplet of water, right? Let me switch over to red. I can do it. There we go. Right, there's a droplet of water. Well, the surface of that droplet is rounded, right? And sort of why raindrops are round, don't fall like pancakes or triangles or any other shape, is because water molecules are a dipole. They have a dipole, right? Water is attracted to water, right? If you look at the structure of water, it looks a bit like this. And because oxygen is an electronegative element, the electrons in the bonds that we've drawn here with these lines, the electrons that are being sort of shared between oxygen and hydrogen spend more time near the oxygen. So the oxygen ends up having a partial negative charge at any given moment. And the hydrogens, by necessity, have a slightly positive charge, right? The electrons are spending more time here, which means they're spending less time here. And so we have, that's called a dipole, right? This separation of charge. Well, guess what? This water molecule likes this water molecule, right? And so on and so on and so on, right? Oop, I already put the positive charge on that. Right? But you get the idea that the water molecules are sticking to each other to a degree. That's surface tension. Well, that's the attraction of water to itself. The reason it plays a big role at the surface is an unbalancing of these dipole attractions, because there's no other water at the surface. This is the air here, if you will, right? Because there's no more water molecules to attach to here, the ones at the surface are being pulled more downward rather than the balance of forces you see on water that's below the surface, right? These guys have a net force downward, which tends to pull the water inward, right, from the surface. Okay, well, that's true in a layer or in a droplet, right? So if you remember that blue line I drew inside the alveolus, it's pulling inward. Our surfactant is reducing this dipole attraction, is breaking up these interactions, and so it's reducing the surface tension on the inside of those alveoli. And that's really important, right? That's important because it increases what's called compliance, lung compliance right here. Right? Lung compliance is simply the ratio of a change in volume to a change in pressure. Think of it this way: the ease with which lungs are distended during breathing, right? If compliance is high, we get a large, top of the fraction here, we get a large change in volume for a small change in pressure, right? It doesn't take much pressure difference to inflate the lungs. That's good, that's what you want. Right? If the lungs collapse, shouldn't say lungs collapse, they can collapse, but if the alveoli and the bronchioles collapse, that means that compliance goes down because we need a greater pressure gradient to cause the same change in volume, right? your change in volume would go down for a given change in pressure. That would be low compliance. That's bad, right? Babies born prematurely lack sufficient surfactant production, and so they have low lung compliance. It's more challenging for them to breathe, and because they're preemie babies, they're very weak, and so they often have to have be put on ventilatory assistance to help them breathe so they don't get exhausted simply in the process of trying to ventilate their lungs. Right? They'll also be given drugs they can be given artificial surfactant. They can be given bovine surfactant from cows. And they're usually given drugs to help accelerate the maturation of the lungs. Okay. Now, Laplace's law is referring to this idea that surface tension can cause collapse, right? And we're going to apply it to the small bronchioles. So we're going to apply it to a tube, right? We're not going to apply it to the alveoli, right? It's a different equation for alveoli. A lot of people apply Laplace's law books, textbooks do this all the time, apply it to the alveoli. Well, the equation for Laplace's law assumes a sphere. What did we just say about alveoli? Are they spheres? Nope, they're flat-walled polyhedrons. So you can't use the different version of Laplace's law to sort of explain this. And so really, it's more about the small bronchioles, this guy, where our collapse pressure, and again, the collapse pressure would be the transmural pressure, the pressure across the wall, right? You can draw that on either side, right? It would tend to make it collapse. And that collapse pressure equals the surface tension divided by the radius of the pipe, right? And so, of course, the surface tension is influenced by the presence of surfactant. And so if we have surfactant, then the surface tension goes down, which means the collapse pressure goes down, and that's good, right? Now, how do we know this is more important in bronchioles than it is in alveoli? Aha, because experimentally, if you take a excised lungs from a dead animal, and you drop the intrapulmonary pressure to the point at which the lung starts to collapse, guess what? You end up with air trapped in the alveoli because the bronchioles collapsed, right? So the bronchioles actually collapse before the alveoli, which confirms the fact that we should use Laplace's law to explain this potential for collapse and the more important reduction in, or increase, if you will, in compliance due to surfactant in the bronchioles. Okay, so yes, alveoli make surfactant, and it's important at reducing surface tension there. But when push comes to shove, and you get to the point at which the lungs or portions of the lungs maybe start to collapse due to either a high surface tension, lack of surfactant, or a low intrapulmonary pressure, it'll be the bronchioles that will collapse, not the alveoli. That's been shown experimentally. Okay. Our last slide for this particular lecture, again, sort of still focusing on anatomy, tying it to the function, is the role of the pleural membranes. Right, so the pleural membranes you have to think of the lungs as sitting in the thoracic cavity, and they're both sitting in a space called the pleural cavity. Right, take the lung out, and you have a space that's called the pleural cavity. So, okay, now stick the lung in, attached to all its blood vessels and the and the bronchi, and you filled the space with this spongy lung tissue, right? Well, one more thing. You have to surround the lung with a serous membrane, which in this particular instance is called the pleural membrane, right? The other serous membranes are around the heart, it's called the pericardium, or around the abdominal and to a degree pelvic organs, where it's called the peritoneum, right? The pleural membrane is the serous membrane around the lung, right? And serous membranes have two layers. They have a layer that is right on the lung. I'm going to draw an arrow here somewhere. Right, so they've got these, see these pleural membrane arrows, right? An inside one and an outside one. Right, so this inside one, this one, is the visceral layer. That is the visceral pleura. This one is the parietal. So you've got a visceral and you've got a parietal pleura. The visceral layer is shrink wrapped onto the surface of the lung. Okay? If you pull a lung out of an animal or a human, you're looking at the visceral pleura, right? You're looking at that surface that you're seeing is the visceral pleura. Okay, now look in the cavity that you just took that lung out of. You're looking at the parietal pleura, right? The parietal pleura is sort of shrink wrapped onto the wall of the space that the lung sits in. So they're actually linked to each other, like way over here where they start, right? The visceral and the parietal are actually linked, attached to one another, right? It's almost like it's one membrane, but you know, one part of it is on the lung and the other part of it is on the inside of the cavity that the lung sits in, the pleural cavity. Now, the two membranes are not literally touching one another. There is a space, this intrapleural space. Here it is. And within the intrapleural space is, if I can do it, fluid. Right? This blue is the pleural fluid. And all the way around, right? That pleural fluid is serving multiple purposes. We'll talk more about it when we get to ventilation, but the pleural fluid, it's sort of it's got a little bit of a lipid component to it, so it's slippery, right? These lungs are sliding up and down on the inside of the rib cage and across the diaphragm all day long, right? So we want low friction, right? So there's fluid in there that reduces the coefficient of friction. In addition, we end up with surface tension that tends to stick the lung to the inside of the rib cage. That's going to be really important when we start talking about ventilation, because there are no muscles in the lung to ventilate the lungs. It is entirely indirect. We actively change the volume of the thoracic cage, if you will, and only because the lungs are stuck to that thoracic cage and the diaphragm below, down here, through that surface tension and another factor, do the lungs track, lung volume track thoracic volume. And that's really important to ventilation, right? The other thing that we're going to talk about that'll be really important is that the fact that the lungs are really elastic and they tend to pull away from the rib cage. The ribs tend to actually pull outward. Their elastic tissue tends to pull away, which means that you end up with this. You end up with a negative pressure in this space. In other words, a vacuum, which kind of like, if you think about it, or you want to think about it this way, it kind of sucks the lung onto the surface of the pleural cavity. And that is really important to avoid the collapse of the lung, right? It's called atelectasis. I don't even know if I can spell that. Right, which is a fancy word for a collapsed lung, right? You want this pressure to be negative so that the lungs stick to the inside of that thoracic wall. If they don't, if that pressure becomes positive for a variety of reasons, the lung will detach and collapse. And then you can change the thoracic volume all day long and the lung volume will not change. You'll have this sort of shriveled lung sitting in here that's not being ventilated. The adaptive purpose of having lungs in two separate cavities. You can collapse one lung and survive. If you only had one lung, well, good luck, right? That's not going to work. So, right, a lot of people survive a collapsed lung because they get to the hospital and it gets reinflated and their other lung was helping them ventilate in the meantime, right? So having lungs in two separate cavities is evolutionarily a really good idea. Okay. I think we're going to stop there for anatomical factors and other, and in the next video we'll jump into, basically into this picture and talk about Boyle's law and ventilation. Okay.